Welcome to the CanMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety and testing. I am your host, Ben Amaralt. I am the Senior Marketing Manager at Medicinal Genomics and proud member of the team that puts on the CanMed Summit. Okay, a few quick announcements to share before we get to this episode. First, we announced our CanMed 26 Advisory Board last week. This year, we have a great mix of familiar faces and new names who will help shape the event by suggesting topics, speakers, and sponsors, as well as review the abstract submissions. You can learn more about each of our advisors at canmedevents.com under the speakers page. But speaking of abstracts, if you would like to present at CanMed 26, our call for abstracts is officially open through November 15th. Each submission will be reviewed by our advisors for novelty, clarity, and the ability to stimulate discussion. If selected, not only will you have the opportunity to present your work to the industry leaders in attendance, but a recording of the presentation will be added to the CanMed archive on canmedevents.com and the Medicinal Genomics YouTube page for additional exposure after the event. Use the link in the show description to submit your abstract today for either an oral or a poster presentation. Good luck, and I hope to see you on the lake. Our guest today is Dr. Hemant Kumar Bid. Hemant is a top educator in the field of biotechnology and medical cannabis, where he uses his extensive background in biotechnology research and cannabinoid research to educate cannabis and biotechnology professionals and infuse the industry with science-based legitimacy. In addition to serving as program director for the Morehouse School of Medicine and teaching graduate students, Hemant also serves as scientific advisor for Essentia Scientific, a company that delivers an array of cannabinoid product solutions, including acidic cannabinoids, with their patented water-as-solvent extraction technology. During our conversation, we discuss what are cannabinoid acids and how do they differ from neutral cannabinoids? Why have cannabinoid acids been traditionally overlooked in research and consumer products? How THCA produces different effects than THC? Why CBDA is more bioavailable and more potent than CBD? How new extraction and stabilization techniques are helping make cannabinoid acids more accessible, and more. Before we get to my conversation with Hemant, I'd like to thank this episode's sponsor, the Cannabis Chemistry Subdivision of the American Chemical Society, also known as CAN. Since 2015, CAN has been building a strong network of scientists in the cannabis space to promote scientific innovation, safety, and development through education, research, and the empowerment of the scientific community. To learn more about what CAN has to offer, please visit their website at can-acs.org. Okay, and without any further ado, please enjoy my conversation with Dr. Hemant Kumar Bid. Good morning, Hemant. Thanks so much for joining us on the podcast.
Good morning, Ben. Thanks for inviting me here, and happy to be here. And looking forward for our interesting conversation about acidic cannabinoids today.
Yeah, like you said, I'm excited to talk with you about the acidic cannabinoids. We recently connected at the Cannabis Research Conference out in Portland, and you were telling me about the new technology that Essentia Scientific is using to extract acidic cannabinoids, which sounds fascinating, and I know we're going to get into that. But first, I thought it would be prudent to talk about how these cannabinoid acids can be used as therapeutics. So maybe a good place to start would be for those who maybe aren't familiar, what are the cannabinoid acids and how do they differ from the cannabinoids most people may have heard of before, like CBD or THC?
Yeah, that's an interesting question. And before we start, just to for viewers that my name is Dr. Hemant Kumar Bid, and I'm a program director for the master's program at Biotechnology and Special Concentration on Medical Cannabis at Morehouse School of Medicine, and also work as a consultant for Essentia Scientific, where we specifically focus on acidic cannabinoids. And that is what we are going to talk about because it is a very interesting topic which is coming again and again in a lot of research these days. So as Ben asked that, so basically that, cannabis plant is very interesting plant and it's produced a lot of cannabinoids and the cannabinoids are stored in the trichomes. So when we think about the biosynthesis of various different cannabinoids in the cannabis plant, so the plant produce the cannabinoids in an acidic form. So cannabinoic acids are the natural precursor to all the cannabinoids like THC and CBD, which are the neutral form. And basically, this all these cannabinoid acids they contain a carboxylic acid group, which is one carbon, two oxygen, and one hydrogen molecule that is removed through a process, chemical process we call decarboxylation with the help of heat. So the main difference in the chemical structure is that most of the cannabinoid acids must be heated to become the neutral forms. And most people are familiar, as when you mentioned, that like CBD and THC, which are the neutral form, but definitely there are a other side of that which is very interesting that like which kind of like give a big curiosity to the scientists and the researchers that like why plant produce the cannabinoids in an acidic form first, and then after that like as I mentioned that there's a decarboxylation process, and then we release the carbon dioxide molecule and then turn into the neutral molecule. So and that's the difference between the acidic form as well as between the neutral forms.
So how does that one difference, how does it affect the biological activity of these
Yeah,
compounds?
that's really interesting question actually. So that like as I mentioned that all the cannabinoic acids that they have the chemical structures, right, which is basically they have an additional carboxylic group which is COH attached to the molecule, and these are the precursor form. That means that these acidic cannabinoids found in the raw cannabis plant when the plant synthesize various different cannabinoids, decarboxylation happened definitely. But so far that we are going to talk about that in detail a little bit, but from last 15 years, I think that there is a more wave and more push about to kind of do a lot of research based on that acidic cannabinoids. And so far, what we know and what we are seeing is that based on the research papers that has been published on acidic cannabinoid, they suggest that the acidic cannabinoid they have their own unique potential benefits, such as anti-inflammatory properties, and they being studied in various different therapeutic effects compare with their neutral counterparts. But one of the unique part that what we are seeing again and again, and recently actually from I think that 7 years ago, John Hopkins published also a paper that acidic cannabinoids have higher bioavailability, more than 10 times higher bioavailability than the neutral cannabinoids, which is like a really a huge advantage for us. And along with that their molecular mechanism is also different, so definitely that there is a lot of difference we can see between the acidics and neutrals, and they can actually go and now these days people are more and more producing new products and actually manufacturing new formulations like Essentia Scientific also, keeping in mind that these acidic cannabinoids have a very high bioavailability, that like to kind of like take the leverage of the entourage effect and create new products, which is really beneficial for the patients.
Yeah, absolutely. I'm can definitely attest to seeing more of the acidic cannabinoid products on the market. And interestingly, there's like usually a combination of the neutral form and the acidic form. So is there some benefit to combining them?
Yes, definitely. There's a lot of benefit to combining them, and along with that, like one of the unique thing, like I said, that the bioavailability, but also they have like a unique different effects, right? So when we think about that, the origin of the both acidics and the neutrals. So acidics, that as we discussed, that it's a raw unheated cannabis plant, that which is present, that because plant produce the acidic cannabinoids first, and then after that we actually heated them, decarboxylate, produce the neutral form, which is CBD. But primary action or the potency, so think about that like maybe acidic cannabinoids, so far the results what we are showing, that significantly more potent for specific disorders like anti-nausea, due to its high binding to a certain receptors. For example, that when we talk about nausea and vomiting, right? So acidic cannabinoids have a powerful anti-nausea effect. The reason being is because that by activating specific serotonin receptors,
Hmm.
and they also have a very impactful inflammation, anti-inflammatory and pain, anti-pain effect, because they act like an NSAID by inhibiting the COX-2 enzyme, that which is kind of like plays a very important role to feel the inflammation and pain in our body. So definitely that like the mechanism is different.
Hmm.
There are a lot of benefit that we are seeing because of its high bioavailability, and along with that, like it can go hand to hand in a very synergistic manner in a good team player with the neutral cannabinoids. So definitely there are a lot of advantage to that, and so far that what we are seeing is that not lot of research or lot of not lot of published papers. The reason being is because there are a lot of limitations, that like there are a lot of challenges we've been facing. So we are going to talk about that.
Yeah, and it's a good segue because that was my next question about why have these cannabinoid acids been overlooked historically in both
Yeah.
research and consumer products until recently, like we just said?
Yeah, it's really a important question to think about as a scientist, as a consumer, that why the cannabinoid acids they have been historically overlooked. As you asked, that like both in the research side, in the clinical side, preclinical research, consumer products, a lot of factors, right? Lot of factors. One of the factor that like I would say that it plays very important role. Being an educator for such a long time in a medical cannabis field, that like I feel like that like education is one of the biggest factor. But
Mm.
along with that, that there are not very good and unique extraction technologies exist that time, that which can provide a lot of good material to researchers to do research on acidic cannabinoids. But along with that, if we compare with the neutral cannabinoids, that cannabinoid acids such as THCA, CBDA, CBGA, they are a little bit more unstable, and naturally they convert to their more well-known neutral counterparts like THC, CBD, CBG when they exposed to heat, light, or oxygen. So this decarboxylation happens naturally over time, and immediately when cannabis is smoked, then like it's decarboxylate. Along with that, that they are like from the beginning, that cannabis being like, as we always talk about, the stigmatize the plant because of its psychoactive bias in research. It
Hmm.
has been major challenge also, because from decades the vast majority of cannabis research folks focus on intoxicating, that like delta 9 THC, which is responsible for high, and like just everything focus on THC. And also that one of the biggest challenge is the regulatory barriers, because of course that like when before 1930 Eli Lilly has the tinctures in CVS and all the drugstores, and then government realized that like okay, it has a lot of potential, let's put in the Schedule 1, which is kind of like really a really sad back for the plant. And
Hmm.
now that what happened, that navigating that complex scenario of the government approvals and regulations and policy made it very difficult and expensive for the entrepreneur, the research, the clinical trials, etc., for acidic cannabinoids to do that right kind of research grade cannabis, limited to the varieties and quantity and strain available to these studies. And also the last challenge was the production and formulation challenge, because of the instability of the cannabinoid acids, make them difficult to incorporate into customer products for long shelf life. So that's also one of the biggest challenge, and these are the all the different reasons that we were not able to research, and we were not able to publish so many papers. But now that slowly the field is saturated, because lot of papers on CBD, as you see that CBD research, THC research, because that was readily available. But we can see now that it's a time to explore more, because we have lot of data on a unique system of endocannabinoid system, how various different cannabinoids interact with various different signaling pathways and receptors in our body. Either it is serotonin receptors, either it is the GABA receptors, and so on. Because so far initially that people thought about that endocannabinoid receptor is just a receptor of CB1 and CB2, and the two molecules anandamide and 2-AG. But that's not the case. It's a whole ocean now. There are so many other receptors are there. There are so many molecules are there. There are so many enzymes are there. So we are kind of moving in a right direction very fast, but definitely more research need to be done.
Absolutely. All right. So maybe this would be a good time to kind of dive into some of these acidic cannabinoids and talk about maybe how they're different from their neutrals, or maybe some of their potential therapeutic effects. I do want to start with THCA, because you made a great point that, unlike THC, THCA does not cause intoxication. So maybe talk about a bit, what makes that difference there, and why that's not the case.
Okay, so yeah, definitely that's we always kind of talk about that the this area of research, or either that the focus of medical cannabis is kind of like uh, either it is consumer, but mostly that then the rec side, that people, it's all about TSC, right? And because of his stigma, because of that the fun part of psychoactive, but that's not the case. TSC, they always say that it's a bad molecule, and I always say that it's not a bad molecule. It's all depends upon that how balance in how much in a balanced way you are using TSC.
Hmm.
TSC is a really great molecule, but in a clinical side that we always talk about, that you need to know about the advantage and disadvantage of any drug XYZ, including medical cannabis, to use it in a very efficient and effective way. So then the question is that, unlike TSC, which is a psychoactive part, TSCA does not cause intoxication in its raw form because of the key difference in a chemical structure. So then when we talk about TSCA, A in TSCA stands for the acid. And the presence of this extra carboxylic acid group prevents this compound from binding effectively to CB1 receptor in the brain that basically produce a psychoactive high. So that's the reason. And TSCA interact with our endocannabinoid system and other biological targets outside the brain. And that's the reason that it creates such a big application. And it's kind of like a bulky molecular structure, which includes an extra carboxylic acid that, as I mentioned, in acidic form, prevents it from the binding effectively to the CB1 receptor in the central nervous system. So that produces the psychoactive effect. So
Hmm.
that's the big difference, that that why it is not high, making the patients high. But rather than acting as a traditional agonist for cannabinoid receptors in the brain, TSCA affects the endocannabinoid system, and kind of like it's interact with various different other pathways, indirect pathways. For example, TSCA is known to inhibit enzymes like COX-1, COX-2, which are involved in their major signaling pathways for inflammation. And this allow it to produce the anti-inflammatory and pain relieving effect without causing high. And also that TSCA interact with TRP channel modulation. That means transient receptor potential channels, which regulate the pain perception and inflammation. One study that was published a couple of years ago, and they actually reported that TSCA was a potent antagonist of the TRPM8 channel. And also that TSCA, there was another paper that they said that it's actually act on PPR gamma, that which is peroxisome proliferator activated receptors, which are the key regulators of the metabolism and fat storage, and that could offer a potential antioxidant benefit and neuroprotective effect. And if you think about that, overall, the regulation of endocannabinoids concentration in the body synthesize that THCA. There were reports that, like they indicated that THCA, the acidic part of the delta 9 THC, may modulate the level of the endocannabinoids, which is produced by naturally in our body and affecting the enzymes to break down, and that basically change overall the activity of endocannabinoid system. But the most important and unique part is because these days the gut microflora, and that research is kind of like in the peak right now, that TSCA, that it influences the gut microbiome and the cellular signaling in the digestive tract, and potentially reducing the inflammation and the regulating the appetite part. But still that, we are in a very beginning phase of this research, and definitely that more research need to be done, that, like to explore exactly the mechanism, and then, what's the value of the TSCA, and how we can create a great formulations combining with the existing cannabinoids.
Wow. So there's a lot there, but one of the ones that stood out to me. So you're saying that THCA can actually sort of affect how the body creates its own endocannabinoids?
Correct.
That's
So,
fascinating.
and again, that, like, we don't know the exact mechanism, but preliminary research is indicating that, and which is really a very important part, because as we talk about that endocannabinoid system is the receptors are expressed in each and every part of our body. And what most of the learners may not know, that, like when body release these endocannabinoids, of course, that, like it creates the balance and homeostasis. That's the basic function of endocannabinoid system. But now there's a paper that which has been published like almost 15 years ago, that these endocannabinoids molecules, anandamide and 2-AG, they play a very important role as a biomarker for so many different female reproductive disorders.
Hmm.
Either it is ovarian cancer, PCOS, ectopic pregnancy, you name it.
Yeah, absolutely. We just the guest I just had, we were talking
Yeah,
about
yeah,
endometriosis.
yes, yep. I saw that.
Absolutely.
Yeah. Yeah. So it was a very interesting that, like, because, like finding endocannabinoid system as a biomarker, which is a big deal, because, like biomarkers can help to detect the disease before time. And, like in clinical side that we always talk about, that sooner the diagnosis, better the prognosis.
Right. So, in theory, then, could THC sort of help someone who has low endocannabinoid tone, or maybe has like a dysfunctional endocannabinoid system to kind of kick it back into gear?
So definitely that it is helping to regulate the production of and synthesis of endocannabinoids for sure, and that's a really a big deal, because that, like the expression of endocannabinoid receptors or either synthesis of endocannabinoids is all depends upon a person's genetics. And that's why we always call that medical cannabis is a personalized medicine, because everybody expression level is different,
Mm.
everybody secretes different amount of endocannabinoids. So the connection and the research, the direction we are moving towards the regulation of TSCA with the production of endocannabinoid system in a balanced way will open a lot of door, but, like more research need to be done.
You mentioned testing for endocannabinoids, and you said that levels in people can be different. How difficult is it to measure endocannabinoids in people? Is it a simple urine analysis? Is it more complex than that?
Yeah, so that's a very important question. Being kind of like working in the field, especially in the analytical field for a long time, I serve as a scientific director for cannabis testing lab in Columbus, Ohio, also for a couple of years. And definitely that, like now that what we are doing is that, like it's very easy to see the expression of CB1 and CB2, because it's just a genetic sequencing, right?
Mm.
So, it's a genomic sequencing, you can see the expression profile. And the another test is that basically that, like either we use the ELISA method, or we either use the GC or HPLC to find that, like the levels of endocannabinoids in our serum. So which is kind of like really a very simple technique, but, like the sensitivity of the technique is still every lab is working on to kind of figure it out, that how sensitive could we go.
Mm.
Because again that, like one of the biggest caveat that, like what we see in the field, because of that, like the affection of the neutral cannabinoids with fats, which is the lipophilic activity that, like when is, let's say somebody consumed that medical cannabis 30 days before and they are driving, something happen on the road and they test it and they found that, like, okay, the metabolized part of 11 hydroxy THC is still there, and now you are going to get fine or go to jail or whatever XYZ, depends upon the policies. But on the other hand, that the good part is that, acidics are more water soluble. So it's a kind of like, like that's the big advantage for the acidics to kind of like do lot of stuff. So definitely there's a more potential to, more for good formulations. And that's the reason that, that when we talk about the bioavailability, we talk about, various different, formulations. It makes such a big difference with acidics compared to the neutrals. But on the other hand, that, before, as I mentioned, that, there were not a lot of techniques were there that which can tell us that, like why, like we can utilize that. But now that, like when I talk with lot of our medical colleagues that who are practicing, Dr. Bonnie, or either, there are a lot of other clinicians that who see the patients, like the pediatric patients with epilepsy, and they kind of claim every time that, like acidic CBDA is more effective than CBD
Mm,
in pediatric epilepsy, which makes a big difference for us to see that, like, okay, it is more effective. And continuously, there are more and more research has been published. This year, 2025, there are two papers came on CBDA and CBGA, and they are very effective in the preclinical model for Parkinson disorders, where they reduce the tau proteins, which is the one of the proteins which accumulate in the brain, to kind of disrupt the normal neural signaling in the brain, and the results are very encouraging. So
mm.
definitely there is a potential in that field, and there's a potential in the acidics. It's all about that, like how much research can we complete within like couple of years. But also that, like one of the also important factor is that, like how much funding we have to do the research.
Right. Always comes back to funding.
Yep.
All right, so I think that's a good segue because we can get into CBDA a bit more now. And if I understand correctly, too, sort of unlike the difference between THC and THCA, where they seem to be kind of acting on different receptors and having different effects, with CBDA, it seems like it retains a lot of the same properties of CBD. It's just much more potent. Do I have that right?
Yeah, so it's much more potent because of that, like it has more bioavailability, right? So that's why that, like it's significantly more potent for lot of different disorders, and act like anti-nausea due to its bind with the certain receptors. So basically that when we talk about that, CBDA, and, like there are a lot of papers that which shows that CBDA to CBD formulations, that it, like when we combined, as we mentioned, that, like combine the acidics with CBD formulations, it's kind of increase the bioavailability,
Mm,
because adding the acidics or adding the CBDA to CBD formulations, it increases the bioavailability of the CBD through several key mechanisms. And the primary mechanism involve the inhibition of the metabolic enzymes, liver metabolic enzymes, which is cytochrome P450, better absorption due to the higher water solubility, as I mentioned, and synergistic effect that boosts the overall impact of the cannabinoids. Now that, let's take one by one. That one is that inhibiting the cytochrome P450 enzymes, which are the liver metabolizing enzymes. So far, the research suggests that CBDA and other cannabinoids can inhibit the activity of cytochrome P3A4 enzyme, which is one of the isomers of cytochrome P450, and a primary enzyme responsible for metabolizing both CBD and many other pharmaceutical drugs. So by
mm,
reducing the activity of this specific enzyme, which is CYP3A4, allow more CBD to pass through the digestive system intact and be absorbed into the blood system. And also that, CBDA has a chemical property that allow it to be absorbed more efficiently in the body than CBD alone. And how it is possible? Because of its increased water solubility.
mm.
Unlike CBD, which is oil soluble, as you know that it is lipophilic, more fat loving, and CBDA has an additional carboxylic group that make it more water soluble, hydrophilic. And this high solubility allows CBDA to be more easily absorbed through the intestinal wall in the bloodstream, where it can be converted into CBD. But some studies suggest that and there are a lot of studies are there now that CBDA may be absorbed more than 10 times effectively than CBD. And the third point is that improve absorption of CBD. Research so far has specifically shown that when taken together, CBDA appears to improve the absorption of CBD. And again, that one study I remember that 2016 they published that and they found that like adding CBDA to CBD more than double the bioavailability of CBD. That's kind of like amazing, right? And then, as I said, good team player, entourage effect, and the synergistic action. When multiple cannabis compounds, we always say the full spectrum, they are taking together, they can produce entourage effect with the help of terpenes and flavonoids, etc. But the presence of CBDA and the other natural cannabinoids in a full spectrum extract, it can amplify the therapeutic outcome of CBD significantly.
Wow.
And that's why CBDA's unique property, like its high affinity for serotonin receptors and its ability to inhibit the COX-2 enzyme, combined with CBD's effects to create a more potent and effective product overall. And the last thing is the prolonged activity. That means some of the CBDS mechanism also that there are papers published that like it's allow the body's own endocannabinoids like anandamide, as you asked the question, that CBDS mechanism that like it allow the body's own endocannabinoids anandamide and 2-AG that to remain active for a longer duration for further increase the body's natural regulatory system and smoothen the neurotransmitter process in the body so much that it is such a fascinating like research outcome that like it's blow my mind every time.
So, if I'm understanding correctly, so CBDA, like, I mean, it sounds like it has a lot of great benefits, but one of the main ones is it's sort of preventing the degradation of either phytocannabinoids or even, I think you said endocannabinoids, right? So that the body is absorbing more of that. So it's sort of just preventing the degradation, right?
Yeah, so basically that it's kind of like preventing the or kind of slowing down or inhibiting the activity of cytochrome P450 enzymes,
Right.
which is responsible for metabolizing CBD or many other pharmaceutical drugs or either many other phytocannabinoids, right? So if we reduce the activity of that
Right.
cytochrome P450, which is a liver metabolizing enzymes, CBDA is allowing more cannabinoids, including CBD, to pass through the digestive system. That means intact, which can be absorbed in the bloodstream. That means higher effect.
Excellent. And so, is the main benefit to combine CBDA and CBD, or is CBDA alone
Yeah, so
beneficial?
that's a really interesting question, and right now that many labs are actually trying to figure it out.
Mm,
That like okay, if we are setting up the experiment, we have actually in Essentia Scientific right now that we have two collaborators in Rome, Italy, and one collaborators in UK. So the investigators that who are doing the research on Fragile X syndrome. So what they are doing is they are trying to do that like okay vehicle alone, and then after that like it's a kind of like a blank group and in the mouse model, and then after that they are using CBD, they are using CBDA alone, CBD alone, and then the fourth group will be CBD and CBDA both combined, because like we wanted to know that like okay what is like, because it's like every disease is so different, every person is so different genetically, that it's very hard to know that who is going to respond how. So it
mm.
is very important to understand the basics, that is CBD is effective in that specific disorder alone, or either CBD and CBDA both are very much kind of like benefited for that kind of disorder. So like a lot of research going on, people are trying to figure it out, but definitely that when we add the acidics in a various new formulations also, it's definitely help and enhance the entourage effect in the body.
Excellent. All right. I do want to move on to CBGA. CBGA is often called the mother of all cannabinoids. What does that mean in biochemical terms?
Yeah, so that's one of my favorite actually cannabinoid. So when we talk about that, like CBGA. So CBGA always and always we call it, like stem cell of all the cannabinoids,
Hmm.
or we call as mother of all cannabinoids. In a simple form, why we called in a scientific world that stem cell, because stem cell has a very powerful, like a cell that which can be de-differentiated into any kind of cells in the body. Huge potential, right? So when we talk about CBGA, call mother of all cannabinoids because it is the foundational precursor molecules from which all the other major cannabinoids are synthesized. So CBGA is basically a starting point in the cannabis plant that, like complex biosynthetic pathway and specialized enzymes that convert it into the acidic form of other cannabinoids. So when we talk about the biosynthetic pathway of cannabinoids in the plant, which happen and finally that kind of these cannabinoids actually store in the trichome, the initial synthesis of CBGA. So the process starts with the combination of the two precursor molecules. One is GPP, which is geranyl pyrophosphate, and another one is OA, which is olivetoic acid,
Hmm.
and the enzyme which is olivitolate geranyl transferase, which is GOT, catalyze their condensation to form CBGA. So there are two molecules, GPP and OA, which makes actually CBGA, and then it happen inside the glandular trichome of the cannabis plant, as I mentioned, the small round balls. And then after that, from there, CBGA is routed into various different branches of the biosynthetic pathway by different enzymes, right? And each enzyme directs CBGA to form a specific cannabinoids. So
Hmm.
what are those specific cannabinoids? Either it is TSCA, either it is CBDA or CBGA. So there are enzymes. One is TSCA synthesis. This enzyme converts CBGA into TSCA, the precursor of TSC. And CBDA synthesis, this enzyme converts CBGA into CBDA, the precursor of CBD. So same thing, CBCA synthesis, that means CBGA change into CBCA, the precursor of CBC. So that's how the whole process goes. But it is really fascinating to see that how one molecule can de-differentiate
Hmm.
into so many different molecules.
Absolutely. And it sounds like it can be used as a therapeutic on its own, right? If it's
Wonderful. Yes, yes. CBGA has a lot of different, like potential, therapeutic potential. And, like CBGA reduce inflammation, specifically, like in any kind of disease. And, like when you, so there was some of the paper that which kind of like claims very well in the past research papers published that, like in acute kidney inflammation,
right.
CBGA may be more effective than CBD because of its potential ability to block specific ion channels and
Hmm.
suppress inflammatory cytokines and inhibit the pathway that CBD only weakly affects that. So if you compare that, with
Hmm.
CBD, definitely in those cases that CBGA is more effective. And the reason is because that acute kidney inflammation, that CBGA actually inhibit it ion channels called TRPM4. So the past research has been shown that the inflammation in the acute kidney inflammation is caused because of the increase in TRPM7 transient receptor potential melastatin 7 channels in the kidney tubular cells. So overactivity of these channels can lead to cell death and kidney damage. But unlike CBD, CBGA is a very potent and effective inhibitor of that channel, which is TRPM7. And by blocking the TRPM7 activity, CBGA can reduce the cell death and protect the kidney during the initial or acute phase of inflammation. But the other mechanism is that CBGA is also a very big suppression of inflammatory cytokines. So if you think about the
Hmm.
acute inflammation, which is basically characterized by increase in various inflammatory cytokines such as TNF alpha, IL-6, and MCP1. There was a preclinical study in 2023 that which found in a mouse model of acute kidney inflammation, and what they claim is that CBGA significantly suppressed the mRNA expression in several inflammatory cytokines
Hmm.
compared to CBD on the other hand was not very effective. And another mechanism is that like CBG is basically inhibit the store operated calcium entry, which is in scientific term we call SOCE. So SOCE is means that it is a store operated calcium entry. It is a mechanism that control the calcium levels and play a role in the activation of pro-inflammatory immune cells. So, research suggested that CBGA has been identified as a potential inhibitor of SOCE in T cells and other pro-inflammatory cells.
Hmm.
And CBD has much weaker effect on SOCE compared to CBGA. So that means the ability to more effectively suppress SOCE is another reason why CBGA has a stronger anti-inflammatory property than an CBD in the early stage of kidney damage. But there are so many different mechanisms are there, and one of the last mechanism I can mention that CBGA can also modulate the inflammatory signaling like JNK and STAT pathway, NF-kappa B pathway, which offer another possible explanation that why CBGA is a strong inflammatory actions. So it's kind of like amazing that like how much research has been done
Hmm.
with CBGA, and and how much we have known that like in various different mechanisms, various different pathways, that how it is important and how that it can affect the inflammatory pathways.
Wow. Yeah, that's fascinating. It seems like it has more of a more specific applications than like a CBD that seems to be a little bit more broad. Is that right?
That's for sure. And also that, like there are papers that which kind of like, like mention that, CBGA's impact or kind of correlation with TRP channels and one of the important cytokine, which is interleukin 2 production. And, and because of these channels and interleukin 2 production are directly correlated with neuroprotective and anti-inflammatory roles.
Hmm.
So that, that's why more research has been done in that area, and literature said that like CBGA has more neuroprotective and inflammatory effect because it is modulating the TRP channels and influencing the immune cell function, which can involve inflammatory interleukin 2 pathways. So there are a lot of different, research has been done, and they kind of propose various different mechanism. And one is that like the mechanism to reduce the neuroinflammation by CBGA is that other cannabinoids and the endocannabinoid system are known to modulate the inflammation by acting on the receptors expressed in the microglia cells, which is the nerve cells, and the brain immune cells. But activation of CB2 receptor, for example, suppress the microglial activation and release the pro-inflammatory cytokine, which reduce the neuroinflammation.
Hmm.
So it can modulate the immune modulation because interleukin 2 is if you think about the T cell growth factor, it can stimulate the glial cell, which is kind of like brain cells in the central nervous system, and it's deregulated IL-2 signaling and contribute to the neuroinflammation. But by potentially influencing interleukin 2 related signaling by CBGA, it can help rebalance the immune response within the brain. So it's kind of like really fascinating that how we are kind of like complementing the different functions and kind of like creating a nuance formulations for specific disorders,
Hmm.
and kind of like giving a value of a specific cannabinoids. And so far that like as we all know that plant has so many different cannabinoids, more than 200 plus, right? But so far that like I don't think that we been researched even more than 10 in a very extensive way because of the regulations, but also because of not having the enough plant material in our hand, in the hand of the scientists. And that's the reason that like we talk about the hybrid plants, right? That how human genome is already cracked down, cannabis plant genome is already cracked down, so we know that which sequence express TSCV or TSCP or whatever, and then we kind of create the hybrid plant overexpress that specific gene so that we can get the more material to do research as a scientific curiosity. But also that every scientific discovery has some sacrifices. So let's say that, like we do TSCP and we have more material, and now we know that TSCV is a anti hunger, that, like we call it skinny wheat, right? So, but on the other hand, that, like, okay, patients are very beneficial with the new hybrid plant, but we play with the genetics, which is against the nature. So that means the plant is weak and plant is not going to survive more than maybe, 5 and 6 kind of like propagation. And then after that, that formulation doesn't exist, and ultimately either patient is suffering, or either that the clinicians has a lot of headache to see that, like, oh, I am not able to provide the specific formulations which was so good for my patients, but that's the sacrifice we have to do.
Yeah, absolutely. All right, I do want to get into some of the extraction technology, because when we first started and we were talking about, why some of these cannabinoid acids have been overlooked. One of the limitations you mentioned was that, the extraction technology or the stabilization techniques just weren't there. So what new technologies are out there and they're sort of helping to do more of this research?
Yeah, that's a really important question, very critical question. I think that, like, we are all are looking for that, and that's why Essentia Scientific is doing such an amazing job, because, like, what we are doing is that we are our the method in Essentia Scientific, that we are using a large scale temperature control aqueous washes to solubilize the acidic cannabinoids from the plant material. So we are basically using the water to leach out the acidic cannabinoids in a very controlled environment, so that we are not disturbing or, providing any kind of opportunity to decarboxylate the neutral acidic cannabinoids into its neutral form, right? So
Mm.
basically, that when we talk about the extraction technology, we have to preserve the acidic cannabinoids, and manufacturers are kind of like, finding various different new techniques to minimize the exposure of heat, light, oxygen, which can cause decarboxylation and degradation. And these all the methods include the advanced low temperature extraction, gentle processing, and innovating the stabilization strategies for the final formulations. So far that there are a lot of techniques that, like out there, and a couple of techniques I can mention to you. One is that cryogenic extraction. So the use of extremely low temperature, such as those achieved with the liquid nitrogen, right?
Mm.
Because cryogenic extraction is basically that liquid nitrogen which freeze, and then that allow for a gentle mechanical separation of the cannabinoid rich trichome and minimize the degradation. But also in some instance, that, like researchers are using cold ethanol extraction. Unlike the traditional ethanol extraction, this method use pre-chilled ethanol at the temperature as low as minus 40 Fahrenheit. And this preserve the acidic cannabinoids and also limit the extraction of undesirable compounds like chlorophyll, which is one of the biggest issue when we isolate that, and waxes to reduce the need for intensive post processing. And also that, like some of the techniques is supercritical or subcritical CO2 extraction, and this method can be used to control the temperature and pressure also. Fresh frozen processing. So this technique also involve immediately flash freezing, and then after that, like using the liquid nitrogen or dry freeze. And this preserve the maximum concentration of cannabinoids and terpenes in their natural form, which is the acidic form. Or some companies are using alkaline leaching. So a greener emerging method recovers acidic cannabinoids by extracting them at a high pH more than 12, and this technique offers very high effective way to process fresh cannabis, avoiding the very lengthy and energy intensive drying process that can promote decarboxylation. There are some new techniques also is coming now. I can see crystallization and encapsulation methods. So these methods actually crystallize acidic cannabinoids within a stable lattice structure, such as crystalline salts and sugar alcohols. And this encapsulation is actually protect the cannabinoids from light, oxygen, and heat, and preventing the degradation. And another method used is a stabilizing matrix. So that means scientists are trying to develop the specialized stabilizing matrix for acidic cannabinoids. For example, that one patented composition use acid and a pharmaceutically accepted solvent to achieve more room temperature stability for almost 24 months. And there are some research that is coming that which is control storage atmosphere so that they can increase the shelf life and that minimize the risk of the oxidation. And some methods are using is improving the drying method. So that means gentle drying technique like vacuum freeze drying are used to preserve the volatile compounds and acidic cannabinoids. And this process actually remove the moisture also. And new methods are coming that which is one of the method I was reading that adjusting the microenvironment pH for certain formulated products. Research has been shown that adjusting the microenvironment pH to an acidic range can significantly improve the stability of the cannabinoids, and it can reduce the degradation of both processing and long-term storage. But also that one of the thing that we need to keep in mind is that mm, purity and potency, consistent purity and potency is the one of the biggest challenge in the cannabis industry. And that's why in Essentia that our method that use the water-based large-scale temperature control aqueous washes to solubilize the acidic cannabinoids, right, from the plant material. And then we sequential course filtration. And then after that, remove the suspension solids and yielding a clarified extract is the key. And then after that we control solution modulation, increase lot of different crude acidic fractions, and then we kind of like finally leach out. And basically that our purity is almost like more than 98%, which is kind of really great. And stability is also really good. So definitely there are a lot of methods are out there. And we are seeing this trend. Lot of methods are coming because slowly everybody is realizing the power and the value of acidic cannabinoids in the plant. And as a scientist, we all are curious what we can do and how we can leverage this unique set of cannabinoids for the treatment of the patients, the welfare of the patients.
Absolutely. And now the end product, is it a sort of like single compound where like it's just a,
So it is very difficult to come up with a like a only the single compound. So there is always a minor amount of the other cannabinoids will be there, but
Mhm.
like it's very minute that like which is kind of like you can say it's a non-significant amount is present there. But now that as you know that like in medicinal genomics that you guys run all the techniques of the testing etc. that the testings and all these machines are so sensitive. If there are a small trace amount is there, it can tell you that it's there. But
Mm.
definitely that like people are working on try to kind of like isolate in a very isolated form, but still we are not there yet.
Excellent. Well, speaking of where we're not there yet, maybe a good place to sort of to conclude things is, what does the next 5 years look like for cannabinoid acids in terms of research or, commercial products that are out there?
Yeah, definitely. That, like, I think that the future is really great because, like, future cannabis formulations are what I can see that we are already started to leveraging both acidic and neutral cannabinoids through a synergistic combinations, right?
Hmm.
Variable ratio formulations, advanced delivery systems, etc. So acidic and neutral cannabinoids, as we discussed, that they affect the body differently, different mechanism due to their unique chemical structure and also target different biological pathway. And that's why acidic cannabinoids are very potent anti-inflammatory, anti-emetic, anti-nausea, neuroprotective. But on the other hand, neutral cannabinoids, psychoactive effect, anxiety, pain relief, etc. So overall, that when we see that like okay, where we are seeing that like it is moving, so definitely we can see the differences. More and more data is out there that where that like we can see that what are the different mechanisms are there, where is the potential that like acidic cannabinoids have more bioavailability is there, acidic cannabinoids are doing really great in antiemetic, right? So it's all about that more knowledge we have, more exploration we have, we can combine this and then kind of like take the leverage as a good team player because we know that like nature has a way to kind of present us and give the indication that why nature put all the cannabinoids and terpenoids and terpenes and flavonoids in one place. There's
Hmm.
a reason to that because they are all good team player. But now that what we are trying to see is that yes, it is there, but can we explore and kind of dissect the various different formulations based on their unique properties, chemical properties, target binding mechanism, as well as their value and therapeutic potential to so many different diseases also. So definitely we are moving that direction, and I can see, as you also mentioned, that like you are seeing in Florida, I see in different states that like more and more acidic cannabinoids are in the combinations, either it is the edibles on oils or either it is the flowers, etc. So definitely we are heading in the right direction.
Excellent. Well, Hemant, thank you again for joining us and sharing all this great information about the cannabinoid acids. Before I let you go, I do want to give you the opportunity to plug any websites or social media or anything where people can get in touch with you or learn more about your work.
Yeah, definitely. That, like, so my email is hbid@msm.edu. So definitely reach out to me. Our program, which is kind of like biotechnology master's program, and especially concentration on medical cannabis, which is a amazing 1 year program completely online. So looking forward to kind of like see if anybody is interested, please reach out to me. If anybody is interested to explore more research on Essentia Scientific, definitely kind of like reach out to me, because we are doing a lot of great things in Essentia, and I'm looking forward to kind of do more research and collaborate with various different groups, so that like we can explore more opportunity and explore more kind of like functionality of acidic cannabinoids, so that we can utilize this unique subset of cannabinoids for patients' benefit.
Absolutely. Great. Well, a great place to find collaborators is at CanMed. So I hope to see you out at Lake Tahoe next year.
For sure, for sure. It is always great to be there in Kanamed. I missed this year, but last year was in I was in Marco Island, and it was always a pleasure to see you and your team there. A lot of great audience, lot of great like posters, and it's a great place to network and kind of like hear out a lot of different like nurse practitioners and MDs and scientists that who are doing a great work. So I think that definitely you guys are doing a phenomenal job, and congratulations to that. And it been kind of like a great seeing you there in Portland, and looking forward to engaging with you in the podcast at Morehouse School of Medicine.
Yep. Looking forward to it.
Okay, thank you. Have a
All
good
right.
day.
Thanks again.
Bye bye.